Hello. The topic is general and medical hematology, type flatworms, class Flukes, Trematoda. Purpose of the lesson is studying of flatworms and their medical importance. And key questions and self-study questions are shown on the screen. The type flatworms includes marine species, freshwater, terrestrial, and the most parasitic species. This is coelomate animals, so they don't have body cavity or coelom around digestive system. They have three true tissue layers, primary germ layers. So the animals are triploblastic. They have the following embryonic tissues: ectoderm, that gives epidermis, mesoderm, that gives parenchyma, and endoderm, that gives gastrodermis. Body of flatworms is usually elongated and slender, leaf-like or long and ribbon-like. Flattened body allows them to still rely on diffusion for exchange of gases, nutrients, and wastes. Epidermis. Free living forms have single layer of ciliated cells. Parasitic forms have syncytial layer that lacks cilia. Muscle layers. Two yellow layers around body wall: sicula muscle and longitudinal muscle. Feeding and digestion. Free living forms are mainly carnivorous. Most species are endoparasites. They have mouth but usually no anus, so they have incomplete digestive tract. Class Cestoda, the tapeworms, have no digestive system. Circulatory system. There is no circulatory or gas exchange systems and no blood. No respiratory system. Nervous system. We see beginnings of civilization. At least some members have decent head. Head with cephalic ganglia, so they have simple brain. Since organs become localized in the anterior end of the animal. They have 1 to 5 pairs of nerve cords connected by ladder-like interconnections. Sense organs. They have eye spots or ocelli, chemoreceptors, auricles. Some have statocytes for gravity and some have retaxes for water current direction. Excretion. Some wastes, like ammonia, are eliminated by diffusion through the body wall. Others have primitive excretory system or protonephridia, tube closed at one end and exiting body at other end. Reproduction. Many reproduce both sexually and asexually. Classification. There are several opinions to classifications of flatworms. One of them we see on the screen. So there are class Turbellaria or planarians. They are free living, they have ventral mouth, and they have locomotion by creeping on cilia and mucus. Class Monogenea. They are ectoparasites on skin, odd gills or fish. They have simple life cycle, so no intermediate host, and they have one generation. Class Trematoda or Flukes or Digenea. All of them are parasitic. And endoparasitic flukes. They are parasites in blood or digestive system. Up to 5 developmental stages in 2 or more hosts. Class Cestoda or tapeworms. All are parasitic. endoparasitics, but long chains of proglottids. Trematoda. Class includes some of the most serious parasites. Almost all are endoparasitic. Adults mainly found in vertebrates. They have leaf-like body shape. They have two suckers for attachment, one around mouth and the other farther down on the body. Adults and larvae inhabit a wide variety of sites in hosts, so digestive tract, respiratory tract, circulatory system, urinary system, reproductive system. Trematodes typically have a complex life cycle with one or more larvae occurring in intermediate hosts and adults in definitive hosts. Adults are typically parasitics of fish or other vertebrates. They have 1 to 5 larvae occur in intermediate hosts and usually a mollusk. Typical life cycle includes egg, usually passes in feces, must reach water to develop. Myracidium, free-swimming larvae, penetrates tissue of snail. Sporocyst, reproduces asexually, polyembryony. Eridia, sporocyst reproduces asexually, polyembryony. Cercaria, emerge from snail, penetrate second intermediate hosts. Metacercaria, juvenile flukes, when eaten by definitive host, develop into adult fluke. Please examine the life cycle of liver fluke or Phaseola hepatica. The standard way to be sure a person is infected with Phaseola is by seeing their parasite. This is usually done by finding Phaseola eggs in stool, fecal specimens examined under microscope. More than one specimen may need to be examined to find the parasite. Certain types of blood tests can be helpful for diagnosis of Phaseola infection, including routine blood work and tests that detect antibodies and immune response. This method is called ELISA. Individual people can protect themselves, do not eat raw watercress and other water plants, especially from Phaseola endemic grazing areas. As always, travelers to areas with poor sanitation should avoid food and water that might be contaminated. Vegetables grown in field that might have been irrigated with polluted water should be thoroughly cooked and should viscera from potentially infected animals. Please examine the life cycle of cat liver fluke, Opistorchis felinus. Ova and parasite stool examination for liver fluke eggs is the only available well for a diagnosis of Opistorchis infection. More than one stool sample may be needed to identify the eggs. So, some preventions. Do not eat raw or undercooked freshwater fish. Lightly salted, smoked or pickled fish can contain infectious parasites. Drinking river water or other non-potable water will not lead to infection with episarcosis. The FDA recommends the following for fish preparation or storage to kill any parasites. Cooking. Cook fish adequately to an internal temperature for at least 145 degrees. Freezing. At minus 20 degrees or below for at least 7 days total time. Or minus 35 degrees or below until solid, and storing at 31 or 35 degrees or below for at least 15 hours, and so on. Thank you for your attention.